Hello, I am Jesper Koll, and I am your host today for this joint webinar on the COVID-19 science. I'm very honored to have organized this webinar in cooperation with the Asia Society Japan, the American Chamber of Commerce, and the International House of Japan. And the topic today will be the science of the current pandemic that is going on. So I'm very honored to introduce you to Dr. Peter Gruss, who is the president and CEO of the Okinawa Institute for Science and Technology, which is one of the world's leading research universities. In fact, it has just been ranked in the top 10 research universities in the world. And, without further ado, I'd like to pass it on to Peter. Please take it away.
Well, good morning, everybody. I'm very happy to have, together with Mary Collins and Mahesh Pandey, the pleasure to entertain and educate you a little bit about the one thing I think none of us has experienced in this way during our lifetime. That is threatening global health, but not only global health, it's also threatening global economy, and it will have a lasting impact. So we believe that we should be able to understand what this is all about and what can be done in the long run. And that is what we would like to do this morning. And I will say a few more words about Mary and Mahesh when they will take over their part of the presentation. So our questions are, where are we, how we got here, and where are we going? Now, these are the key points, and I think this is not a surprise. We need to know what is causing COVID-19, what is the virus, where's the virus from, how can infection be prevented, how can we analyze infection? Even more important, how and when can we develop a vaccine? And then something that is very puzzling to many of us, why do people respond differently? I think we are all aware that the high-risk group is not the children. The high-risk group are the people above 65 or 70. So what can we learn from other countries? Because given the experience in other countries where the outbreak has been earlier than in Japan and Okinawa, respectively, we do have some insights what they did, and we can compare to what we did. Well, last couple of points, second, third waves, when will the virus disappear? Will it ever disappear? And if that is not the case, what can we do in the meantime? So this is the very last point, scenarios for a new normal. So let me just say that the infection and infectious diseases are not new. Infectious diseases have been always with us. As a matter of fact, we can call it, they have a co-evolution with all the organisms. So also then, communicable diseases existed during humankind's hunter-gathering days, but they took off very much so once the humans started to cluster. So the first communities, agrarian communities were founded about 10,000 years ago, and that was heaven for a microbe. Malaria, tuberculosis, leprosy, influenza, smallpox, and other first appeared already 10,000 years ago in those communities, and they could be transmitted between communities. In Japan, for example, in the Tenpyo area from 735 to 737, about 1/3 of the Japanese population was killed by smallpox, a virus that, of course, has been a plague for all over the world. Bacterial diseases like cholera, tuberculosis, typhoid fever, ulcers, respiratory infections, food poisoning can be easily treated with antibiotics. Why? Because the bacteria are so different from the human cells. So there are many drugs that can specifically attack the bacteria because the bacteria have biochemical pathways and structures that can serve as a target for the antibiotics without actually interfering with the health of a human cell. This is quite different with viruses because viruses are much harder to treat. Why? Because viruses, if you look at the virus literally in a droplet, viruses are not a living organism. The virus needs to survive to enter a host cell. In our case, it needs to attack a human cell in order to replicate. And this is something you all know which has been overlooked by President Trump, because he has thought he made a smart remark by using disinfectants that can indeed dissolve the outside membrane of the virus, but he clearly overlooked that the same disinfectant will also dissolve the membrane of the human cells. So you drink this disinfectant, you will die because your cells will die. It's a little minor issue, but it tells you about the educational level of a current US president. Now, the Spanish flu is thought to have infected around 1/3 of the world's population and killed at least 50 million people. It is the deadliest pandemic in modern history. Now, another flu, it's the called the Hong Kong strain. The Hong Kong strain appeared about 68 and lasted until 69. The Hong Kong strain has killed 1 million people. We are currently, Mahesh, you may have the correct number, we are currently about at 300,000 dead people by COVID. So what I'm trying to say, we all need to be careful, but we have to also put it into perspective. So without microorganisms, we would cease to exist. That's also a fact. 37 trillion cells are in our body, and about the same number is in our gut. We need it in order to digest properly our food. We need it to make beer. We need it to make dairy products. We need it to bake bread. We need it literally in most of our daily lives. Some bacteria play one role or the other. And this is just for you to get a feeling what this world is like, the world of microbes. There are more microbes in a teaspoon of soil than there are people on Earth. Viruses also emerged at a very early stage of life. As I said, they co-evolved with living organisms. And there are about 10 million viruses in every drop of surface seawater. Now, don't be afraid. These viruses are not harmful for humans. They are viruses that are attacking mostly bacteria. Now, similarly, the coronavirus, as we call it properly, so coronaviruses are also ancient. They originated about 55 million years ago, and they are closely associated with the evolution of bats and birds. Now, these viruses are quite different from a human cell, and you can see there is this is probably not visible for them, right? There is in the center of this spherical object, you see in reddish color, this is the genetic material of the coronavirus, and the genetic material of the coronavirus is RNA. Our genetic material is DNA. Now, if you have RNA, you need a very specific, I don't have it here, but you need a very specific polymerase that is able to replicate that RNA. Now, that polymerase is prone to generate mutations. So mutations occur during replication, but they are stochastic. And they are only subsequently being recognized if there is an advantage to one of the zillions or trillions of normal viruses, and one in a zillion or billion could have a mutation that then gives that one particle an advantage, for example, by jumping the species barrier. So that's the way it goes. It's classical, evolution. And here, this is just a photo that gives you a depiction of what we are talking about in size. You can see the big object on the left is the tip of a needle. That's the tip of a pin. You see the orange colored structures. These are bacteria. And you can see the bluish structure are cells, and the reddish little dots, these are viruses. So viruses are 500 times more tiny than a normal average human cell. That gives you an idea, what are we talking about? And what I like to do, because I found this was such a remarkable, informative video, that I think the next 5 minutes, I will be very good for everybody to see and get an educational insight.
This is SARS-CoV-2. It belongs to the family of coronaviruses named for the crown-like spikes on their surfaces. SARS-CoV-2 can cause COVID-19, a contagious viral infection that attacks primarily your throat and lungs. What actually happens in your body when you contract the coronavirus? What exactly causes your body to develop pneumonia? And how would a vaccine work? The coronavirus must infect living cells in order to reproduce. Let's have a closer look. Inside the virus, genetic material contains the information to make more copies of itself. A protein shell provides a hard protective enclosure for the genetic material as the virus travels between the people it infects. An outer envelope allows the virus to infect cells by merging with the cell's outer membrane. Projecting from the envelope are spikes of protein molecules. Both a typical influenza virus and the new coronavirus use their spikes like a key to get inside a cell in your body, where it takes over the cell's internal machinery, repurposing it to build the components of new viruses. When an infected person talks, coughs, or sneezes, droplets carrying the virus may land in your mouth or nose and then move into your lungs. Once inside your body, the virus comes in contact with cells in your throat, nose, or lungs. One spike on the virus inserts into a receptor molecule on your healthy cell membrane like a key in a lock. This action allows the virus to get inside your cell. A typical flu virus would travel inside a sac made from your cell membrane to your cell's nucleus that houses all its genetic material. The coronavirus, on the other hand, doesn't need to enter the host cell nucleus. It can directly access parts of the host cell called ribosomes. Ribosomes use genetic information from the virus to make viral proteins, such as the spikes on the virus's surface. A packaging structure in your cell then carries the spikes in vesicles which merge with your cell's outer layer, the cell membrane. All the parts needed to create a new virus gather just beneath your cell's membrane. Then, a new virus begins to bud off from the cell's membrane. Now, with the virus spreading in your body, how can you develop pneumonia symptoms? For this, we'll have to look into your lungs. Each lung has separate sections called lobes. Normally, as you breathe, air moves freely through your trachea or windpipe, then through large tubes called bronchi, through smaller tubes called bronchioles, and finally into tiny sacs called alveoli. Your airways and alveoli are flexible and springy. When you breathe in, each air sac inflates like a small balloon. And when you exhale, the sacs deflate. Small blood vessels called capillaries surround your alveoli. Oxygen from the air you breathe passes into your capillaries, and then carbon dioxide from your body passes out of your capillaries into your alveoli so that your lungs can get rid of it when you exhale. Your airways catch most germs in the mucus that lines your trachea, bronchi, and bronchioles. In a healthy body, hair-like cilia lining the tubes constantly push the mucus and germs out of your airways, where you might expel them by coughing. Normally, cells of your immune system attack viruses and germs that make it past your mucus and cilia and enter your alveoli. However, if your immune system is weakened, like in the case of a coronavirus infection, the virus can overwhelm your immune cells, and your bronchioles and alveoli become inflamed as your immune system attacks the multiplying viruses. The inflammation can cause your alveoli to fill with fluid, making it difficult for your body to get the oxygen it needs. You could develop lobar pneumonia, where one lobe of your lungs is affected, or you could have bronchopneumonia that affects many areas of both lungs. Pneumonia may cause difficulty breathing, chest pain, coughing, fever and chills, confusion, headache, muscle pain, and fatigue. It can also lead to more serious complications. Respiratory failure occurs when your breathing becomes so difficult that you need a machine called a ventilator to help you breathe. These are the machines that save lives and that medical device companies currently ramp up production for. Whether you would develop these symptoms depends on a lot of factors, such as your age and whether you already have an existing condition. While this all sounds scary, the push to develop a coronavirus vaccine is moving at high speed. Studies of other coronaviruses led most researchers to assume that people who have recovered from a SARS-CoV-2 infection could be protected from reinfection for a period of time. But that assumption needs to be backed by empirical evidence, and some studies suggest otherwise. There are several different approaches for a potential vaccine against the coronavirus. The basic idea is that you would get a shot that contains faint versions of the virus. The vaccine would expose your body to a version of the virus that is too weak to cause infection, but just strong enough to stimulate an immune response. Within a few weeks, cells in your immune system would make markers called antibodies, which would be specific for only the coronavirus, or specifically its spike protein. Antibodies then attach to the virus and prevent it from attaching to your cells. Your immune system then responds to signals from the antibodies by consuming and destroying the clumps of viruses. If you then catch the real virus at a later stage, your body would recognize it and destroy it. In other words, your immune system is now primed. Collecting evidence on whether this will be possible, safe, and effective is part of what's taking researchers so long to develop a vaccine. It's a race against time to develop a vaccine amid a pandemic. Each step in vaccine development usually takes months, if not years. An Ebola vaccine broke records by being ready in 5 years. The hope here is to develop one for the new coronavirus in a record-breaking 12 to 18 months. While all this will take time, stay home if you can, to protect the most vulnerable. And don't forget to wash your hands for at least 20 seconds and as often as possible. This video was a collaboration between Nucleus Medical Media and the What If channel, where we usually dive into hypothetical scenarios about the human body, humanity, our planet, and the cosmos. Check out our channel by visiting the link in the description below.
So this gave you an overview of the entire infection process, and it gave you also an idea that at some time in the future, we will have a vaccine available, but it is unclear yet when that is going to happen. There are fast tracks that think about sometimes next year, probably towards the end. If we are unlucky, it will be much later. So people are, of course, discussing alternative treatments. And here is a brand new piece of data that has been generated in my old institute, in Max Planck Institute for Physical Chemistry, where they have crystallized and found this particular structure here on the right-hand side, which is the protein that is necessary to replicate the RNA genome. And you see the genome of the virus is this helical bluish structure. And if I now start the movie, you see single nucleotides coming and are being polymerized based on the template. So it's a mirror image of the blue that is now generated in red. So why am I showing you this? Well, this type of protein is highly specific. It is otherwise not available or present in human cells. So it constitutes a perfect target for a drug that may be able to inhibit this particular polymerase. The first drugs are available. They are not very effective because they could not be targeted specifically. And you've all read about remdesivir from Gilead or Avigan from Fujifilm. Both actually can interfere negatively with the RNA, so-called RNA-dependent RNA polymerase. But with this particular insight of the molecular and atomic structure, we now can design molecules that specifically attack this RNA-dependent RNA polymerase from the coronavirus. And I can share with you that the structure of that is different from other RNA polymerases from other viruses. So here we have a chance by combining, let's say, empirical studies like with remdesivir with modeling studies to come up with a much better and more efficient drug. So now we will move to Mary Collins. Mary is the provost and has been and is an immunologist and virologist. She has directed from 2005 to 15 the Medical Research Council Center for Medical Molecular Virology at University College, has worked with viruses, viral vectors for clinical use and gene therapy. And we are very happy that Mary is here since 2016 and is now the provost for all our academic activities. So Mary, wait, I think this we do later. Okay.
Thank you very much. Pleasure to talk to everybody. So I'm now back in doing some virology, which is great, actually. There's a good news in the pandemic for me in some ways. Okay. So I'll talk a bit about testing for coronavirus. Japan has adopted this strategy of identifying infected people, these marked in red here, by a so-called PCR, polymerase chain reaction test, and I'll explain that on the next slide. And those people are tested based on symptoms plus risk factor identified by a doctor. Then their contacts are traced, and those contacts are then tested to see if they have the virus. And in Japan, all infected people, however mild their symptoms, are then isolated in hospital or hotels, sometimes adjacent to the hospitals. And this kind of approach works very well at times of low infection. It's not so great when a peak is ripping through a country, but before the big surge of cases, or after, before another wave, for example, this test isolates strategy is working. And this slide just shows what a PCR test is. We have the virus RNA, as explained by Peter. We make a DNA copy, and then we copy those copies. And so every cycle we go through in this little reaction in a test tube, we get a doubling of the amount of material. So simple calculation, one virus can be detected after, say, 40 cycles. We have 1 trillion copies. And we can detect one virus in an inoculum here. And no virus gives no copies, which is wonderful. So it's highly specific, sensitive, and accurate. And at OIST, we've set up this testing as part of Okinawa. And we've been commissioned by the prefecture to do this diagnostic test for COVID-19. And we're using a WHO recommended method that's spread around the world. We receive samples from the most remote hospitals, basically, from Hakubu, which is in the north, and the Yaeyama Islands of Miyakojima every day. And they're either throat or nasal swabs and sputum. And at the moment, we're 6 hours from receiving the sample to giving a positive or a negative test. The tests look like this, they're wonderful. So these are some control samples, in fact, from the OIST lab. Here, we have about 1,000 copies of virus. The number of cycles you need to go through to amplify something detectable tells you how much virus there is. This is about 1,000 copies, and this is about 100 copies of the same test sample. So it's highly sensitive and specific. And we're planning to develop, we're not doing our own development really, we're implementing methods that are identified around the world. We'd like to be able to do this faster. There are some tests related to PCR that seem to work in under an hour. Using saliva is going to be a lot easier because we can get people to self-test, drop some saliva into a tube, deliver it to the OIST testing lab, and we can automate this so that we have a higher throughput. And this sort of testing, I think, will be with us in the very long term. So we need to make it efficient, accurate, affordable, in fact, also. The other thing that Peter has been talking about was antibodies. So the PCR test is okay as long as you are infected by the virus. And the PCR test will detect people who are infected something between 2 days before they start to show fever and symptoms to maybe 10 days after infection. But really, to establish how much infection we have in the population, we need to measure antibodies, because these are stable and they remain, as far as we know, for several months at least in people. So this is a better way of seeing who has been infected in a population. It's a stable measurement. So we're implementing, again, an antibody test that was developed at Mount Sinai here. And we're commissioned by Okinawa Prefecture again to survey patients from 5 hospitals. And that will actually give us a number of how many people in Okinawa have been infected by COVID-19. In this case, we can obviously think of better tests, and we can modify the test. It'd be quite good to be able to do it from finger pricks of blood. So again, we can ask people to self-sample. There have been a lot of news about this home kit format. Looks a little bit like a pregnancy test, and you can test yourself. Most of those have been sadly inaccurate so far. So I'm always a bit worried about an inaccurate test. If you base your behavior on on a result that's slightly random, then you actually may change your behavior in a bad way. So I think at the moment we're on a lab-based test for now. And a lot of money has been raised for us by the OIST Foundation in our US-Japan series of webinars and concerts, because these tests are not basic research. They're just things we have to do at this moment, and the things we have to carry on doing, I think, for the rest of at least my working lifetime. Okay, so the future of COVID tests, they'll be with us for years to come. We can have a very fast genome test. We probably want to correlate this with mild symptoms so that we don't test everybody all the time. We want to trace contacts by phone apps. That's a cool thing to do, definitely. And we can then immediately ask everybody who's been in contact to get tested. There are pinch points in life where we may want to test people who don't have symptoms. These could be travelers, PCR tests, for example, Olympic visitors, Olympic athletes, OIST visitors. These are all possibilities to allow us to open up to more normal behavior. The antibody tests tell you really who's been infected in the population. At the moment, as Peter said, we don't know if antibodies protect you. If we do get some data that that's true, then we could use these tests to look at healthcare workers, teachers, travelers, to see if they are protected against infection and they will never be infected again. So I think smart tests are with us for a long time. Antiviral drugs or vaccine. This is really a global problem, and the solution needs to be affordable and deliverable worldwide. So antiviral drugs can lessen disease, mainly in the developed world. Optimal testing worldwide is going to be needed for a long time. This is really the pinnacle of people's battle against viruses. This is the elimination of smallpox. This was done by rolling out a very effective vaccine. And you can see it took a very long time worldwide because you have to find everybody on the planet and immunize them. And this kind of vaccine eradication needs amazing logistics. It needs a cold chain usually to develop the virus, to deliver the vaccine that's to everywhere in the world in an active form. And it also needs people to preferably respond well after one shot of vaccine, because actually immunizing people 3 or 4 times, if you've found them once, is not very easy. So smallpox was eradicated. I'm optimistic, but we need to be careful about what we promise, I think, in terms of vaccine. And COVID-19 vaccine, many excellent teams are working on this. Virus is not mutating substantially at the moment. It has random mutations, as Peter said, but they don't seem to be that highly selected in the population as it moves. So I think that unlike HIV, for example, which mutates incredibly rapidly, even within one individual, this virus does not mutate at that rate. So I think that's good news for a vaccine, actually. The virus doesn't persist. It's not like HIV, which is a persistent viral infection. So if you protect somebody, then they can be protected against, they can be cleared of virus. Notes of caution, how to test. If the virus epidemic goes down considerably, we may be having a problem of where to test this vaccine. And there have been suggestions that volunteers could be vaccinated and then challenged with the virus. That's not a trial I would do myself, but we would need young and healthy people to do that. You also want to ask, how long will protection last if we vaccinate people? Do we all need to get this annually like a flu shot? There is one, slightly, again, worrying note is the vaccine can enhance inflammatory diseases. And this has happened for dengue fever or respiratory syncytial viruses, where the vaccine has actually made the disease worse in some people. And that is, I think, my only negative about the prospect of having a vaccine. And with that, I will hand over.
Thank you very much, Mary. And it's a great pleasure to introduce Mahesh Pandey, who is an associate tenured professor at OIST. Mahesh was trained in computer engineering, got his PhD from the University of Pittsburgh, followed by a postdoc at Los Alamos National Laboratory and Harvard University, and has been the author of some excellent and top-ranked papers. And today, he's also here, not just because he's an excellent scientist, but because he's also assisting the research teams at the World Health Organization's headquarters in Geneva to help model the global spread dynamics of the COVID-19 virus. So, Mahesh, here you go.
Thank you, Peter. Thanks, Mary. Good morning, everyone. Peter's and Mary's presentation has focused at the molecular scale and the testing aspects. I want to proceed further from the molecular and cellular scales and its implications at the population level. As Peter mentioned early on, viruses have been with us for a long time, and viruses regularly jump from animals to humans, and roughly at the rate of 2 viruses per year, at least since the 1960s, as we see in this plot over here. There are 3 reasons why the cumulative number of viruses is flat before the 1940s. First is that the viral detection tools themselves matured between the 1940s and 1960s. So it is not that there were no viruses being transmitted from animals to humans prior to this period. The second is lifestyle improvements have translated to increase in wildlife trade, thereby increasing the risk of these viruses making a mutation that allows them to jump from animals to humans. The case in point here, the specific example is the Wuhan seafood market, which is considered the source of the current outbreak. One remark I would like to make here in this context is that actually, if you look at the statistics from the CITES database, global database, the wildlife trade in the United States is actually 10 times larger than the wildlife trade in China. So the risk is, in fact, if we just take that statistic, the risk is actually higher in the US than it is in China. But there is a very strong correlation that has been observed, one-to-one correlation between the lifestyle improvement, improved lifestyle to the increase in wildlife trade. The third reason is basically with the increase in global population, which caused forest deforestation due to encroachment for agriculture. And we know the first Ebola outbreaks appeared at the forest farm perimeters in Africa. This is proven now from this study in 2017. You see these images, these triangular points, the top, so the top image here is from 2004. The bottom is for the same region in Africa from 2014. And these triangles were the areas where the perimeters existed as of 2014 when the outbreaks were reported. Unfortunately for us, there is, of course, a lot of richness in viruses across the animal kingdom. And unfortunately for us, animals that are rich in viruses are also ones that humans come in regular contact with. These are bats, primates which are closely related to us, mammals, which are also closely related to us, rodents and domestic animals. So there is that added risk for that reason as well. Now, coming to the viral transmission at the population level, let us suppose that we start with a population that is uninfected, as shown in black here. Let's say the virus makes a jump to one human being and infects them, shown in red. Then the virus starts ramping up and infects more and more people. And as the infections rise, as shown in blue here, through the exponential increase, the number of infected people exceeds the number of uninfected people, which is shown in black. Then the recoveries start to begin. And once the recovered people are immune for some period, then we see what we refer to as the flattening of the curve, either due to the immunity or due to other protections we take. So what might they be? So let us consider one way to approach the risk involved. An oft-quoted number, R0, is something you might have come across in media, which is the average number of people that are infected by a person who is already infected by the virus. If R0 is greater than 1, then you see an exponential rise in the number of infections. And if R0 is less than 1, the infections are falling. Media quotes this number only, but it alone is not enough. If you look at the plots to your right, the percentage of asymptomatic infections is equally important because it tells us what proportion of the infected people is shedding the virus and infecting others without being detected, without showing the symptoms. To give an example, if you look in this plot, SARS, COVID-19, and HIV, all three of them are not in a similar range, between 2.5 and 5. Yet SARS was not as widespread as the current COVID or HIV because all the symptoms the onset of symptoms occurs at the same time as the viral shedding occurs. At the other extreme, we have HIV, which has near 100% asymptomatic transmission. COVID is around 30 to 50% from the cases we have seen to date. So keeping this in mind, a near-term risk reduction as we wait for vaccines to be developed, first and foremost is personal hygiene, which you saw in the video earlier, because it is within our individual control. The second is social distancing, which is the most important because speaking from personal experience studying the pandemic dynamics, the most difficult locations where to do contact tracing or to track hotspots is train stations, airports, and that is where, or public places in general. There is this notion of herd immunity that has been mentioned. It is a dangerous concept. We have seen it in Sweden, for example. But if you also, you'll also see that the proportion of deaths reported by Sweden relative to its neighboring Scandinavian nations, which enjoy the same level of high quality healthcare, proportion of deaths is higher. So there is a grave risk involved in going for the idea of herd immunity. And finally, I want to end with the slide with the advice that we need to test, and test. That is the only way out for us, because without the numbers, without the statistics, we are utterly clueless. Many of the questions that arise about why Japan behaves in one fashion, whereas Europe or US are showing different dynamics, is because we don't have enough numbers in some of the countries. And to give you an example of the importance of social distancing, here is an AI-based tracing app that was conducted at a beach at Fort Lauderdale during the spring break. This was after the US was in full-blown COVID crisis. During the spring break, after stay-at-home orders went into effect, there were people on the beach. Taking their cell phone pings, this company did this AI-based tracing and asked how many of these people on the beaches ended up elsewhere, and how, under the assumption that they were infected, or even a small proportion of them were carrying the virus, how far could it have proceeded. So that is the beach. You do not see many orange dots to your right, because that is the sea. And we have selected a bunch of orange dots along the beach. And now if we zoom out from the beach and ask, where do these orange dots show up, and how many other orange dots, how many other cell phones are these orange dots coming in contact with, it quickly spreads across Florida, across southern United States, southwestern United States, and is approaching East Coast. So it is that dangerous. Finally, the long-term risk reduction. As Mary and Peter have pointed out, the vaccine, if possible, will be available likely in the order of 2 years. The chances of us getting a vaccine are perhaps better than in the case of HIV. But can we stamp out the virus? It would require extreme measures. As Mary pointed out, smallpox took a long time. The other aspects in long-term risk reduction is improvement in healthcare. We have observed a very strong correlation between the quality of healthcare and the mortality rate. Prepare for future waves. Something that is not repeated in the media, you'd see in the bottom right corner, the plot over here. The first wave is always more devastating. It is stronger because you have a large population pool that is uninfected. But later waves, despite having some level of herd immunity, will always linger longer. And that must be taken into account, especially considering the effects this has on the economy. So prepare for future waves. Control wildlife trade. I think that is a no-brainer. And control deforestation. And if you look at the deforestation, actually, Asia has unfortunately done a great job of deforestation. The high risks, if you look at the plot at the bottom left, the high risk area is actually the Amazon forest, South America. So that is where the most deforestation and human contact with wildlife is occurring. And furthermore, what are the questions? It may take about 2 to 5 years, to find answers to many pressing questions. Why is there such huge variability in how infected people respond to the virus? Why is there a huge variation in the detected viral particles? The plot you see above shows the viral load varies from few particles to 100 million particles per milliliter. And it seems to be constant from toddlers through senior citizens. We don't understand why. How long does the immunity last? Does the COVID-19 depend on climate? How fast does the virus mutate? We think it does not mutate as fast as the HIV, but we will need to wait and see what new statistics tell us. So there is a lot more left to learn. And with that,
thank you very much. Jesper, this would then be the, say, educational part, and maybe we can take a few questions now. And if the time permits it, we would have then a few add-ons regarding what could be new normal. What could be our life? What could our life look like in the years to come?
No, I mean, absolutely fantastic. Thank you very, very much, for this wonderful lecture. I think we all learned a lot, about the science. I want to stay focused a little bit on the science and, open the floor for some of the questions that we've received here. The first one is sort of a very sort of straightforward one. I mean, is this actually a strong and efficient virus? It's a question from Mike Alfond. And, he's asking, how much exposure do I need in order to get affected? Is it just one particle landing on my face, or do I need to be coughed upon? Is this a shotgun virus, or is it a sniper virus?
I can take that one. One particle is enough. But to come in contact with that particle, being coughed on is a pretty good way. I think the droplets in the air are worrying people more than virus remaining on surfaces. It's quite a fragile virus, so there's some studies that show you can detect it on surfaces, but it's not clear if it's really infectious after some time on a surface. One is enough, but if you're exposed to a high load of virus, for example, you're a healthcare worker, then your disease may be more rapid and more severe. So, but one is certainly enough to get it.
Right. Excellent. Another question, comes from William Hall, which looks at, is there any evidence that different gene mutations give you a higher risk of mortality or not? When there's this discussion about, some of the Latin gene mutations, having a higher exposure, maybe that could explain why the mortality rates in Asia, tend to be lower, at least for the current data, tend to be lower than what we've had. Any progress there on gene mutations being linked to the eventual mortality of the virus?
Well, there are two levels I see to answer that question. First is the population genetics. And the population genetics, unfortunately, is not available yet in a volume or in a breadth that is necessary to address that question. So what one would essentially do or need, one would have to have quite a number of genomes that come, let's say, from Italy, and compared with genomes from China, compared with genomes from Japan, for that matter, in order to address that question properly. That's the first. So we need simply more population genetic analysis in order to address that question properly, which now can be done, and one could maybe even focus on those patients that have suffered particularly badly from the virus. But the second level is also an issue that, let's say, we have some insight, but not a full understanding, and that is the individual level. Why do some individuals respond differently from others? We all know that, statistically speaking, the children are very mildly suffering from a virus infection, while a high-risk group, including diabetics, including cardiovascular issues, including obesity, those people are highly vulnerable to a virus attack. And we know that the virus, in order to enter a cell, needs that specific lock, the receptor that you've seen in the movie. And that receptor is called ACE, ACE number 2. And as it happens, in older people, or in some people, let me just say, in some people, the number of receptors in the heart tissue is higher than in other, let's say, younger people. So we do get a first glimpse what could cause the vulnerability, but we are far away from getting a holistic understanding why some people die and others don't.
Great. Excellent. There's a whole cluster, if I can use this word, a cluster of questions. You know, what obviously stands out just, you know, in an amateurish way, looking at the mortality data, you know, obviously Japan, you know, sort of stands out as having a very low one. You know, can the three of you sort of offer your insights? Is there any explanation? What is the reasoning behind that?
Oh, I'll take my go at it and you can disagree. That's what science is about here. So certainly in Okinawa, there are very few infections. That I'm sure about, okay? Because we've been participating in PCR testing and probably hundreds to thousands of people have been tested recently here and we haven't picked up any new infections. So I think for a reason I don't understand, Okinawa, at least in Japan, has very few infections. And I have a feeling that some of the mainland may be the same. Mahesh.
What I would like to add to what Mary has said, the early onset dynamics we saw in Japan as well as in Okinawa, the fluctuations of the infections followed what we call Poisson statistics, which is random infections. The cluster infections, the hotspots, as that we speak of when we are looking at the exponential growth that we saw starting from February 16th for Japan, and much later in Okinawa, that has flattened out. So my feeling is it is very important to run antibody tests across the Japanese population to get a good idea of what was the real scale of infections. Because as Mary's plot showed, the PCR tests might have petered out, the efficacy might have petered out because the Japanese infection curve has flattened out more or less.
Great. In that same vein, as you know, there's a lot of speculation about the BCG inoculation, you know, which we do get here in Japan, that the BCG inoculations have had somehow some positive impact.
I'll take that one. I think that's a Donald Trump type assumption, okay? I feel there's no scientific basis for that.
Okay, that was a quick answer. You know, I mean, it seems. Look, I mean, this is obviously a very, very complex, you know, an incredibly complex situation that's unfolding here. And from listening to the three of you, I think it's going to be at least, you know, another 12 to 15 months. You know, before we actually have, you know, the consistency of all the data to be able to make some more affirmative statements about things. And so, Peter, maybe we can, you know, I may ask you to just speculate very quickly a little bit about, you know, the you know, moving on from the science to daily life. I mean, when are we, you know, what the new normal coronanomics? What do you see as some of the shifts that are going to impact all of us?
Well, first, I admire the terminology that came from a bright economist in Tokyo, coronanomics, that Jesper has published in the Japan Times. And what I have here is, before we can think about doing anything seriously, we have to know that the transmission is controlled. So what Mahesh has suggested, you need for every infected person, that this person infects less than one on average. Then we can call it the transmission is controlled. We need to have the hospital system working and being ready for the next wave, or if you get, like Mahesh also said, regional clusters, vulnerability settings, elderly homes, mental health facilities. You've read, I'm sure, about the elderly homes in the United States, where literally hundreds of people died away because they weren't careful, or rather the means weren't there to protect them. Workplace preventive measures, physical distancing, that has to be, and we are going to do that in OIST, hand washing, respiratory etiquette, and so forth. I find very much, say, promising what we have here as a tracing app. Tracing app, even in countries that are extremely careful with giving away personal data, are now being introduced, and what they would allow is that as soon as somebody who has that app comes close to somebody who is infected, the app will warn you, so that you have an information that you should, maybe be tested, or at least you understand that during the course of a day, you were in close proximity to a virus positive person. Communities should have a voice very clearly, because, communities have to continue to live. So now, what are the lasting shifts? Well, currently, I think there are two big areas. One is cushion the downfall, and the second is accelerate recovery. So cushion the downfall. We can see that many governments internationally, to one of the, or some of the bullet points I have listed here, some will nationalize, some will take equity stakes, some will provide loans, others will choose to regulate. We've seen in Japan and other countries, there is helicopter money for all, albeit at a low level. But there are stimulus packages and say key points I've outlined here. Accelerate the recovery, and this is the long-term impact. We'll have 4 targets: globalization and trade, technology and innovation, societal impacts, and behavioral shifts. Automation, additive manufacturing offer promises to, let's say, relax the global supply chain. And we all know this is one of the key issues that is a problem in running economy during times of crisis. So it may actually lead to businesses moving from offshoring to nearshoring or even reshoring of production. This is, in fact, in essence, it's boosting anti-globalization trend. And it is an issue to either renationalize or regionalize the points. Technology and innovation. I believe social distancing, video conferencing, virtual classrooms, and telemedicine, digital commerce will be with us forever, although not at that magnitude. Next generation working technologies could invest in augmented and virtual reality, I think will be very helpful. Governments are deploying surveillance technologies. I was mentioning this. You have to be very careful. If I look at the app that voluntarily people use in Germany, that's something different as opposed to what the Japanese government does. Surveillance technologies. Researchers can, work on technologies that would even allow more complex mechanics to be generated nationally with 3D printers and so forth. Societal impacts. Of course, the safety net has been a big issue if you look in particular in the United States, in part in the United Kingdom, where not enough tests and beds originally were available. So we have critical points: health coverage, gig workers, jobless people, or if you look at it in a positive sense, Generation Z. So the guys that are being born into this post-crisis, they never experience anything else. So for them, life would be normal. So I think behavioral shifts, same thing, in-person connections over social media, remote shopping, trust currently, trust again, or trust in national governments in many countries, not all, but many, is low. Polarization is high. I don't know if you heard about that there have been 10,000 people that want to demonstrate against the lockdown in Munich. The officials only could allow 1,000 because of the spacing. And that will happen this weekend. I don't understand how people think, if they think at all. And this is in part also true for xenophobia that is actively being promoted by populist governments. And with this, I thank you, and I was very happy to be with you. I don't know if we have more time for questions, but in any case, we are available. Thank you all.
Great, Peter. Thank you very much. I certainly, share a lot of the anxieties, and potential problems that you've got there. And obviously, the world is a very different place. Whether you've got 5% unemployment or whether you've got 25% unemployment, the threat of real xenophobia, scapegoating, etc., is very strong. Let me end on one final question, for you as scientists. In terms of global cooperation, data sharing, there's a lot of criticism, particularly from the US administration on the World Health Organization. Is global cooperation at the science and medical level, is that actually still working? And what would you do better?
I think I can attest to the fact that the cooperation across the global scientific community has actually increased manifold. I'll give you specific examples where people chose to, usually when we do some research and come up with a new result, we want to keep a tight lid on the results until we publish or patent them. But now the results are coming out on what we call the preprint servers. People share them freely. Data is being shared freely. Universities across and research institutions, clinics are sharing data, and they're sharing know-how. Whatever they learn, they're sharing patent-free, royalty-free. And I think it is heralding a new openness in the global scientific research landscape. I can show you one example of this interactive website about the test on how the virus got out of Wuhan. And this data came from China, from all countries that the virus got into. And it also is a prime example of how to be careful about social distancing. So we know stopping travel, we thought would have stopped. It did not stop the Black Death in the medieval times. It did not stop the coronavirus. We know the virus is known to have centered around the Huanan seafood market in Wuhan in late 2019. 4 cases grew to dozens by the end of December, but we now know from that there were nearly 1,000 asymptomatic cases, as a result of which each patient was infecting. The R0 value I mentioned was 2 to 3, not less than 1. And this is data from the Chinese cellular networks that showed the travel from across China. And from Wuhan on Jan 1st, there were 175,000 people departing in preparation for the Lunar New Year. And about 85% of those we know were infected, and they were seeding new clusters across cities in China. This was as of Jan 21st. And 2 days later, the authorities locked down. So you see as an abrupt slowdown in the travel, but the clusters were already seeded and they kept growing. This was Jan 21st, but the outbreak had moved across China. So now I'm moving from Chinese data to worldwide data. And we know that at least the first known detection from Wuhan was to Bangkok, a 61-year-old lady, but that was the first detected case. We know that the ex post facto, there are asymptomatic cases that have come up now from late December in France. So there were cases coming up in Seoul, Taipei, Tokyo, Sydney. And then once the lockdown goes into effect in Wuhan, it was too late because the new clusters had already been seeded. And now we are looking at the data from across Europe, Iran, and despite the fact that China had started testing systematically and led to a lockdown, the clusters had reached US. And on March 11th, President Trump suspended travel across Europe, but it was too late. So you can see this kind of data would not have been possible in a pre-COVID-19 era. We are looking at data that has come from all sorts of governments with all styles of governance, freely sharing data to make it possible to understand what happened here.
Thank you.
Wow. Wow, that looks very impressive. Can you just, there's just one question that came in from Catherine Sasanua. Why was China able to contain the spread so radically?
Well, it's the form of governance they have. It's simple.
So it was just the
That
harshness
is
of
about the most diplomatic answer I've ever heard.
Okay. So it was the harshness of the lockdown, was it?
Yeah.
Right. Okay, I think we've run out of time. Thank you very, very much. This was really a very enlightening event. If you do have follow-up questions, please don't hesitate to either contact the American Chamber of Commerce, the Asia Society Tokyo, or the International House in Japan. We'd be very happy to put you in contact with the OIST people. Thank you very much, Peter, and your team. Thank you.
Thank you all for listening. Thank you for your interest, and have a good day.